All right, so we're going to get started. And today our first speaker is Jim Mitchell. He got his PhD from Colorado State University in 1978. And he came to NCI in 1979, and he's been here ever since. He's chief of the radiation biology branch, and at times he's chief of the radiation oncology branch as well. So he's had a lot of experience, and I'm sure you'll enjoy his lecture today. He has a physical problem, so he's going to be lecturing from the chair. Jim, take it away.
Thank you, Terry. I so much apologize because I normally like to stand up when I talk, but finally my age is caught up with my white hair. I have hip issues, and about 3 weeks from now, we're going to correct that, I hope, with some surgery. So please bear with me. I'm going to try to get you through this and while sitting. I apologize for that. So I'm a radiation biologist. As Terry mentioned, I've been here forever. I'm a survivor, working on year 35 at NIH and NCI. What I want to do today is talk a little bit about free radicals, but with a different spin than what you've heard in previous lectures. I'm going to focus primarily on radiation, because that's what I do. And I don't get to publicize radiation very much. People are always afraid of it. They don't want to know anything about it. So you've got to bear with me, and hope you'll learn a little radiobiology this afternoon. So you don't need to be a physician to look at this CT scan through the lungs and notice that there's something wrong here. Like there's a huge mass right here. This happens to be a non-small cell lung cancer. In the United States this year, there will be 224,210 cases of lung cancer, both small and non-small cell, and 159,260 deaths. You won't see that on the evening news tonight, but that's a significant number of people. And so what do you do when you encounter a patient like this? No doubt there have been numerous patients, even today, presented with a CT scan like this, which is an inoperable tumor. Well, the best option for this tumor is to treat the tumor with free radicals, and that is ionizing radiation. So I want to take you through this scenario. You will see this slide coming up in subsequent slides, and I will pretty much end with this slide of this CT scan. So what do I mean with free radicals? Well, we're using ionizing radiation. There are several ways that radiation interacts with matter. Depends on the energy of the incoming photon. It's either the photoelectric effect, which is just enough energy to knock off a fast electron from an orbit of a molecule, or a little bit higher energy, Compton, where the incoming photon knocks out a fast electron and then is scattered so it can go and knock out other electrons and other nuclei. Both processes, energy absorbed is converted to fast electrons. So when you irradiate anything, photons interact with matter and they release fast electrons. It's the fast electrons that's doing the damage. So the radiation beam is non-discriminating. For a given radiation field or volume, free radicals are generated uniformly in both normal tissues and tumor. That's what I mean by non-discriminating. You set the field, you irradiate, everything in that field is exposed to free radicals. It's unlike chemotherapy drugs that you inject into a vein, usually, and they distribute through the vasculature and hopefully make it to the tumor for cancer treatment. But there are a lot of barriers along the way. There are no barriers for radiation. You set the field, you turn on the machine, you irradiate, you deliver the dose and the free radicals. The free radical reactions that occur as a result of radiation exposure are very quick. In fact, by the time the machine turns off, most of the free radicals have done their damage, okay? They're very short-lived. And so we talk about, I'll have a timeline for that in a minute, but there's two ways that radiation interacts with tissue and matter, and that is by direct action and indirect action. Direct action is about 20% of the given radiation dose damages cells and tissues this way, and it's simply just knocking off fast electrons, which can break chemical bonds, and these bonds aren't repaired. You can have permanent damage. The bulk of the radiation damage is inflicted via indirect effects, and that is the interaction of radiation with a very simple molecule called water. In fact, if you irradiate a class of water, there's about 16 different products that can be formed very quickly, and some of these interact with each other and dissipate back to ground state. But water radiation can yield a variety of radicals, a hydrogen radical, OH radical. Superoxide can be generated by interacting with an aquated electron that's kicked off from water. And notice the time scale. These are all formed in less than a second, far less than a second. So the formation of these products are in spurs and diffusion of radicals out of the spurs. And so we're basically dealing with these radicals, and this, of course, is not a radical, but it has the potential to become one when it's reduced by iron to OH radical. And again, all this is happening very quickly. Most of the damage imparted by radiation is via indirect effects, primarily because you're sitting there now, and about 80% of you is water. And this particular target for radiation-induced damage, which we now have known now for many years, is predominantly the DNA molecule. The DNA molecule is a setup for indirect effects of radiation because, as you remember from your biochemistry, it has a hydration shell wrapped around it. So any interaction in this water shell around the DNA and you form an OH radical, it has enough distance to diffuse the DNA and cause a variety of DNA damage points. Most likely one that leads to cell killing is a DNA double-strand break. So this is a direct action. Indirect is through an intermediary with water in this case. For the most part, what imparts most of the damage from ionizing radiation via indirect effects is the OH radical. It's an extremely bad player. It's got a short lifetime. In chemistry terms, it's what we call diffusion control. It interacts with pretty much whatever's in front of it and on the order of 10 to the minus 9 seconds. It has limited diffusion, only 2 nanometers. So if you look at this hydration shell, it's around 4 nanometers. So if an OH radical is formed in this hydration shell, it can diffuse enough to hit the DNA molecule and cause damage. It's a strong oxidizing agent. It breaks chemical bonds. It can induce lipid peroxidation and a whole host of other bad things. The OH radical is not a nice thing to have around biology. And of course, you can get that from radiation, but you can also get it from reduction of hydrogen peroxide to OH radical. Here is the timeline again, the time scale all the way down to 10 to minus 3 seconds, the events that are occurring, OH radicals near the target, which I call a BIM, a biologically important molecule, which we believe to be the DNA molecule. And so you get into secondary radicals. You get chemical repair in some cases. You can actually have a radical on a important molecule like DNA, like a DNA carbon-centered radical. That can interact with oxygen and give it an enhanced radiation effect. We'll talk about that in a minute. When you come down to seconds, you've registered the damage. You have some oxidized DNA or other molecules in the cell, which can lead to strand breaks, which can obviously affect proliferation and lead to cell death. And that's why we use radiation to treat cancer, because it kills cells, okay? Unfortunately, if these damaged sites are not repaired correctly, you can also lead into mutation and carcinogenesis. And perhaps you can also see in tissue late effects, which my former boss many years ago said, who was a radiation oncologist who took care of patients. Fibrosis, scarring, vascular damage is the gift that keeps on giving, because you can get a patient through cancer treatment, get a complete response to their tumor, but 2, 3, 5, 8, 10, 15 years later, they come back with normal tissue damage via fibrosis. At this intermediate time here of seconds to a few hours to a few days, there's a whole host of things going on within cells and tissues, which I call physiological and tissue effects. Host cell activation, cytokine induction, gene expression, signal transduction. And a lot of these are very interesting areas to intervene to decrease the radiation damage. And there's been a plethora of work done in the last 5 to 10 years on small molecules that inhibit, various signal transduction pathways and so on, either to enhance the radiosensitivity in tumors or protect against the damage in normal tissue. So radiation obviously imposes oxidative stress. I've gone on to say that it's probably the ultimate in oxidative stress. It's really cool in inducing it. And so you get all these free radical products and peroxide to boot, reactive oxygen species. Is any hope of surviving it? And the answer is yes. The cells and tissue have devised ways over many years to protect themselves against oxidative stress. And some of the systems that give us some of that protection are enzymes like superoxide dismutase, which converts superoxide to hydrogen peroxide. Glutathione, a central redox molecule in cells, provides a lot of reducing equivalents. It's present in cells and tissue around a millimolar, even higher in the liver. Glutathione peroxidase, which can detoxify peroxide, catalase, and enzymatic repair systems, which I'll talk about in a few minutes, and perhaps others. And of course, the best way to protect against radiation is distance and lead. I would prefer distance, over lead, but so radiation inflicts a lot of damage and it can cause a lot of bad things. Let's talk about glutathiones because I spent a decade of my life on that molecule, so I want to talk about it for a bit. I made this cartoon because that was my granddad's pocket watch, and I thought about the interactions of gears and mechanisms in a pocket watch, much like biochemistry reactions, going forward and backward and that type of thing. But glutathione, as I said, is a central redox molecule in the cell. It's synthesized via cysteine, which is a very neat amino acid, thiol-containing amino acid. And it can detoxify peroxide, and superoxide obviously fits into this. It's also kind of cool in getting rid of drugs and xenobiotics through glutathione S transferase. So it's a neat way of detoxifying unwanted agents in cells. And it's obviously involved in the pentose pathway, glucose metabolism. It's really an interesting molecule. And so this is some of the things that it can do that I just mentioned. In the 1980s, we capitalized on a biochemist from Cornell named Alton Meister, who made a lot of neat compounds that could either inhibit glutathione synthesis, or he had some agents whereby you could increase glutathione in cells. So we studied those, this compound buthanine sulfoxamine or oxythiazolidine, and asked the question, if you take glutathione out of the cell, would you enhance the sensitivity to radiation? Or conversely, if you elevated glutathione in a cell, would you protect against radiation? Okay? Now, there's not a whole lot that's going to interact with an OH radical. I once had a molecular biologist tell me that there was enzyme systems that detoxified OH radicals. That is the most ridiculous thing I ever heard of. OH radicals react with whatever they please, okay? Whatever they please. They don't need an enzyme. The diffusion control. When we're trying to interact with radicals generated by radiation, we're really after the secondary radicals. Can we do anything with those? And I would kind of gauge peroxide as a secondary radical, particularly if it's reduced to OH radical. So we did a series of studies looking at this one. This is a survival curve of cells exposed to different doses of radiation under aerobic and hypoxic conditions. I'll come back to why I'm keen about hypoxia in a few minutes. But if you expose cells under aerobic conditions to radiation, you get a survival curve that looks like this. This is old school clonogenic assay. Even though our laboratory is one of the first to publish the use of colorimetric MTT assays, you heard of those? In fact, we have a paper in Cancer Research in 1987. It's been cited more than 3,500 times. It's not cell survival. It's cell growth. If you want cell survival, you do clonogenic assay like this one's here. And so you see, if you treat cells with BSO and you take glutathione levels down to less than 5% of control, you radiosensitize a little bit, but not much. There's no error bars there because they're the size of the symbol. I like to repeat experiments a lot. So there's about a 8 to 9% enhancement of radiosensitivity. To be honest with you, I was very disappointed. I thought it would be more than this, and it wasn't. And then I got even more disappointed when we put cells under aerobic conditions and we increased glutathione. We doubled the amount of glutathione in the cell, and absolutely nothing happened under aerobic conditions, no protection whatsoever. So I don't want to say I wrote off glutathione, but I kind of wrote off glutathione back many years ago as a major modulator of radiosensitivity. Some, but not much. Under hypoxia, the story was about the same. A little enhancement under hypoxia, but not much happened at all with oxythiazole. Made a little protection, but not much. So under aerobic conditions, when you irradiate, you have radiation forming these radicals, perhaps on the DNA molecule, that interacts with oxygen, and they actually can fix the damage. Now, I'm from Tennessee, so you notice a southern accent. So whenever we say we're ready to do something, we say we're fixing to do this, right? Meaning we're going to do it. But when I say fix damage here, I mean fix it so it's not repairable. So oxygen is a bad thing to have around a radiation beam, okay? Because it fixes damage permanently. There are not many ways to repair an organoperoxide on a DNA molecule. Under hypoxia, though, if you take the oxygen away, these endogenous reducing agents, like thiols, can chemically repair this damaged molecule and restore it. That's why you have more resistance under hypoxia than you do under aerobic conditions. In fact, for X-rays, the ratio is about a factor of 3. Okay? So cells are 3 times more resistant to radiation under hypoxic conditions. You'll see why I'm interested in that in a bit. In the 1990s, when we were finishing up this glutathione work, I met this guy named Dave Wink, who was all keen about nitric oxide. And I'm sure you've heard Dr. Wink speak already in this series of lectures. And we were all keen about nitric oxide back then in regard to radiation response, particularly of hypoxic cells. And so we actually used an agent called DINO. Perhaps David mentioned that. It's a nitric oxide releasing agent that you can use in aqueous conditions. And we found if you increase the concentration of DINO, that is, you let nitric oxide into the medium and the cell environment, as you increase the concentration, you could bring this curve right back down almost to aerobic conditions. There's a way in radiobiology we look at that called a K-curve. So nitric oxide is almost as efficient as oxygen in radiosensitizing hypoxic cells. We found it to be a great hypoxic radiosensitizer, but difficult to administer in tumor-bearing animals because nitric oxide has a tendency to drop blood pressure in mice. It makes it a little bit difficult to deal with blood pressure loss and keeping an animal alive. But we looked at a whole series of these nitric oxide releasing agents, and they all work quite nicely in enhancing hypoxic cells. So I got to tell you now why I'm hung up on hypoxia. That big whopping tumor you just saw, that non-small cell lung cancer, there is a significant presence of hypoxia in that tumor. Because of altered blood flow in tumors, you get regions where there's very low oxygen concentration. That affords resistance to radiation. So in radiobiology and radiation oncology, for decades now, we've looked at means by which we could enhance hypoxic cells to radiation. So you see why we're so keen in nitric oxide and other agents like that. So back to this patient, how is this patient treated with free radicals? Well, normally in non-small cell lung cancer, you give fractionated radiation daily, Monday through Friday. I always make the insulting joke about my radiation oncology colleagues that they don't treat tumors on Saturdays and Sundays because they must not grow over the weekend. I asked my boss once, I said, how come you don't treat on Saturday and Sunday? Because my tumor cells in the lab grow continually. And he said, okay, Mitchell, don't be a smart aleck, right? Actually, that's been studied. There was a European group who actually studied treating radiation all the way through, all the weekends for a couple of weeks rather than breaking it up into weekly fractions. And there was some interesting results. I don't have time to go into that. But normally in the United States, Europe, other countries, you give about 1.8 to 2 gray. If you're not interested into the new terminology, it's rad. We used to use a dose of radiation as a rad. 200 rads, 100 rad equals 1 gray or 2 gray, okay? And that's given over 7 weeks, about 35 fractions, so a total dose of 63 gray to this tumor area, okay? So important factors to consider: the dose, of radiation you give, the fractionation, how the dose is divided, the time of treatment course, and the volume of tissue. We've got it down now so it's very simple. It's the 4 R's of radiotherapy: repair, repopulation, reassortment, reoxygenation, and radiosensitivity. These two, repair and repopulation, are vital for normal tissues. These are more important in regard to the tumor response, and we'll go over those. So, radiosensitivity and tolerance. I had the privilege for 30 plus years of teaching radiobiology to radiation oncology residents. And one of the first things I asked them when you see a tumor, a patient's tumor coming in on scans or whatever, how many cells are there? And so I asked the question of a 1 centimeter tumor, which you can see on MR, almost see on an X-ray. How many cells are there? 1 centimeter. That's pretty small. Half an inch, right? There's 10 to the 9 cells in a 1 centimeter tumor, okay? It's a huge number. So people get excited sometimes when they're looking at a new chemotherapeutic agent or another agent that gives them what's called a partial response, a PR. The PR, though, is a decrease in tumor dimension by 50%. Not much of a response. A CR means the tumor disappears for some length of time, but it can disappear and not be visible with 10 to the 8th cells present. So just because you treat and the tumor goes away doesn't mean it's gone away for good, okay? So if you consider chemotherapy, and I think you all know the most of the chemotherapy, cytotoxic chemotherapy drugs that are used, if you give 8 cycles and you get 50% cell kill per cycle, that's 0.5 to the 8th power, or 0.004. If you have a 10 to the 9 tumor, that'll still leave 4 million cells. Okay? Pretty humbling, okay? If you use radiotherapy, you give 30 fractions, you get 50% cell kill for each fraction. That's 0.5 to the 30th power. That's 9 times 10 to the minus 10. That will pretty well take you down to less than a cell. So you might have a chance, assuming all those cells in the tumor are aerobic and not hypoxic, okay? So what if the cell kill is only 20% rather than 50%? My goodness, 0.8 to the 30th power is only 1.2 to 10 to the minus 3. You leave a lot of cells around. So the inherent radiosensitivity of the tumor cell is another major factor in how this tumor is going to respond. We have been interested in using sensitizers for a number of years. That is, something that will give you an enhancement of radiosensitivity. So if you got a twofold enhancement, you go 0.8 to 0.4, then you'd have perhaps a tumoricidal survival. The bigger the tumor, this is in centimeters, the more dose it requires to get local control. This was published in 1980. This has been known a long time. And just about a year ago, I heard someone say, well, there's no dose response for radiation. And it says, what do you get when you have gray hair and you've been around for 35 years? You can go back, 30, 40 years in literature and say, you mean you didn't read Gilbert and Fletcher? Radiosensitivity and tolerance. I had a privilege in the 1980s of working with John Minna's group, who was isolating a lot of lung cancer tumor lines from patients. And the sensitivity of lung cancer varies a lot from variant small cell adeno all the way down to classic small cell. Look at the difference in radiosensitivity. It's quite large. Unfortunately, though, there's not much difference in the inherent radiosensitivity of most normal tissues and tumors. You can kind of see that from this plot right here, where you kind of have the extremes. This is a human cell line from patients with ataxia telangiectasia, mutation in ATM gene, so they're very sensitive to radiation cell killing. This is bone marrow from mice and humans for that matter, mammary cells, crypt cells. Their radiosensitivities all fall pretty much in range with this, so there's not a lot of difference. The tumors that we have the hardest time treating are these two. Actually, I could add a third one here. I don't have it. But glioblastoma, brain tumors, and melanomas have a very high surviving fraction at 2 gray compared, for example, to lymphomas. So you want to get a really good response, treat a leukemia and lymphoma with radiation. It melts away. Okay. But you treat a brain tumor with radiation, it does not. Pancreatic cancer also winds up in this category as well, as being not very responsive to radiation. The tolerance doses for various normal tissues, so you got a problem. You want to treat this tumor, but you got normal tissues all around it. Sometimes the tumor wraps around normal tissues. And so invariably normal tissue is in the radiation field being treated as well. So what's the tolerance? We talk about this in terms of tolerance doses in 5% of patients in 5 years. So if you want 5% of your patients to come back with complications, you give the TD5 dose. So for heart and great vessels, about 60 gray, esophagus the same. Lung is fairly sensitive, 17.5. Spinal cord, you really have to worry about spinal cord in regard to radiation response. You can't go more than 47 gray for 20 centimeters of spinal cord in the field. And skin is not really an issue because of the high energy machines that we use, unless you want it to be, unless the tumor is very superficial. So, but the tumor control may be anywhere from 65 to 70 gray. So you got a real problem. You need 70 gray in a tumor, but you got normal tissues experiencing problems at doses less than that. So it's a real complicated problem of how you get this differential. And so there have been people like myself interested in radioprotectors. Crazy as it may seem, would it be possible to identify an agent or an approach that would give differential protection to normal tissue and not to tumor? Hard to come up with that. Or would there be a radiation sensitizer that would sensitize tumor and not normal tissue? In oncology, it's the game of, balances, tumor to normal tissue. It's the same for chemotherapy. It's the same for molecularly targeted drugs. They're all the same. Are you getting a differential effect in tumor and normal tissue? Now, radiation oncology has come about addressing this issue by using fancy physics and advanced techniques. I'll have something to say about that toward the end, to maximize the dose of the tumor. So the goal is to get a therapeutic gain effects in tumor compared to normal tissue. Can tissue repair? The answer is yes, thank goodness it can. The problem is tumor can repair too. There is some evidence over the past probably 20 or so years to show that normal tissues, at least in animals, tumor-bearing animals, normal tissue can repair better than tumor. But that's not to say that tumor cannot repair. It can. A lot of this work was done in regard to repair by doing a split-dose survival curve, as shown here. You take cells in a petri dish and you give them a dose down that kills one log of cells. Then you wait for 8 hours, and then you re-expose to radiation. And you ask the question, does this curve continue exponentially, or do you repeat the shoulder here? And as you can see in this graph, you repeat the shoulder, meaning that over that 8-hour interval from one radiation dose to the other, the cells have had the capability to repair radiation damage. The kinetics of this has pretty much been demonstrated in a variety of cells and tissues. The half-time for repair is about 20 to 40 minutes. Normal and tumor cells can repair damage. In the clinic, sometimes we give 2 doses of radiation a day, and we always try to make sure that there's at least 6 hours in between those doses to allow for complete repair in normal tissue. It's called BID, twice-a-day radiotherapy. Some drugs can inhibit this repair and may enhance both tumor and normal tissue, and the major lesion that's repaired is a DNA double-strand break. And there's just been an explosion of studies now in DNA repair pathways. The most important two for radiation is homologous recombination and non-homologous end joining. There's a very complex configuration of repair entities that come together to repair DNA breaks. Some of the really neat work done on this campus by Bill Bonner, who came up with a gamma H2AX studies where you can actually see foci after radiation that identify DNA double-strand breaks, and with time they go away, showing repair. Some beautiful work. These are the foci that you can get with gamma H2AX. If you don't know about this, I would really encourage you to read some of this literature. It's fascinating. Is a tumor tissue hypoxic? That is a problem that's been around for we've known about it at least 50 years. We've not done much to address it, but certainly you can have hypoxia, that is low PO2 in tissue. For example, how many of you grow cells in the laboratory? Yeah, pretty, a lot of you do. And so what do you think the oxygen tension is of normal tissue in you right now? Let's just say muscle. What is the oxygen concentration, or the percent of oxygen in that tissue? How much? 5. Pretty good idea. That's a pretty good value. And what are you growing your cells at? 20. You're exposing your cells to oxidative stress, almost an order of magnitude higher oxygen concentration, right? So I always slap myself. I do it too, okay? I did take off about a year and made a glove box and tried to do stuff at lower concentration before all the fancy incubators came along. And certainly you can alter the redox status of cells by growing them at lower PO2. There's no question about that. And if you're trying to set up tumor lines for patients, you'll have a lot better success if you take the tumor suspension from the patient and put it in a low PO2 incubator rather than one at 20%. At any rate, hypoxia is a problem, and it's because of the abnormal vasculature in tumors. So who cares about it? Well, we've already talked about this resistance factor to radiation. It's also true for cytotoxic chemotherapy drugs. A lady by the name of Beverly Teicher, who's now at NCI, has been here the last 2 or 3 years, published a paper back in 1990 showing that you could also get resistance to cytotoxic chemotherapy drugs by these various factors because of the presence of hypoxia. Not from the mechanism that we're talking about with radiation, but simply the fact that you don't get the drug to the tumor cells because of the altered blood flow. It's something that I'm not criticizing the young generation, but a lot of people now in the cancer biology lab think that a tumor is just a log phase bag of cells growing with all they need, oxygen and nutrients and that type of thing. The fact of the matter is tumors are train wrecks. I mean, they have survived and they figured out ways of surviving under very abnormal conditions. And so this issue of getting the drug to the tumor cell is a major issue. And it's also a major issue if you're going to be thinking about molecular targeted agents, small molecular weight agents as well. Can we measure the oxygen concentration in tumors? Yes. Back in the 90s, there are a series of studies using an Eppendorf electrode. It's actually a needle that you can actually just stick right into tumors. It had a computer-driven backing device so it could kind of work its way through the tumor and make multiple measurements. And so here's a frequency distribution of PO2 in normal breast, fairly high PO2, but in a tumor you can see very low in some areas with no oxygen at all. Now this was used to do some studies like these, and this was kind of a classic paper published in 1996 by Huckel. It really started a revolution, so to speak, of molecular biology in regard to hypoxia. Because he looked at cervix cancer patients prior to treatment, and he measured the PO2 before they were treated with radiation, okay? And then he looked at the overall survival probability at some point out to about 80, 70 months. And you can see that patients who had initial oxygen levels less than 10 millimeters of mercury did less well in regard to survival than patients who had higher oxygen levels. But an interesting thing is, the same was true for surgery. And to my knowledge, there's no free radical mechanism associated with a Bard-Parker scalpel, okay? But what hypoxia demonstrated in these patients was the preponderance for recurrence and metastatic disease. And so this study here, which was a clinical study, started a search for molecular components that might be responsible for this. And this was the beginning of papers on HIF-1 alpha, hypoxia-induced factor alpha. It was started from this study. Head and neck cancer, same thing. If you have low PO2, disease-free survival is much worse compared to higher PO2. Why isn't it all patients have hypoxia? I don't know the answer to that. But for a given tumor type, about 50% of patients will present with low levels, and 50% higher levels. Interesting that it works out that way, but that's the way it is. Implying that you really need, if you're going to customize treatment in regard to hypoxia, you really need a better way of doing it than oxygen electrodes. Non-invasive imaging would be preferable. And so I encouraged many years ago my colleague Murali Cherukuri, Murali Krishna, to begin some studies looking at possible ways of measuring PO2 non-invasively by imaging. And so he's using a technology that's related to NMR called electron paramagnetic resonance. He's been developing instrumentation, contrast agent, and spectroscopic concepts for several years now. And now in a mouse, he can actually look at a normal mouse. This is an MR with an overlay of the PO2 map and a tumor, an animal bearing a tumor. And you can see here's the tumor with very low PO2 in this part of the tumor. He uses a contrast agent, which is a proprietary agent by General Electric. It's called a Trittle radical. It has very nice spectral qualities in regard to EPR imaging. It also, by the EPR, we get nice penetration, several centimeters in tissue. This compound is fairly non-toxic, and it gives a beautiful signal when no oxygen is around, which is really great. And as you add oxygen, you decrease the height and wavelength, bandwidth of this signal. And here's a calibration curve for PO2. So he's now generating some very beautiful images of an animal. Here's a healthy animal looking at their PO2 of the normal muscle. Here's a tumor, and you can see there are regions in this tumor that are very hypoxic right here. And so there's the PO2 levels. He can actually do serial imaging across only in about 1 to 3 minutes with resolution about 1.5 millimeters. And you can see as you cut through the tumor, the oxygen profile is different. Not only is it different, but it's changing. If you look at certain areas in the tumor, like this position number 1, you can see at 4 minutes it's at about 35 millimeters mercury, but by 10 minutes it's down to 0. And then it can go back up and oscillate. Whereas there are some regions like region 4, you can follow that across here. It stays very low. We call this chronic hypoxia, and this is transient hypoxia. And so if you're going to combat this problem clinically, you've got to address both of them. There's another issue about why we fractionate radiation treatment in the clinic. One is for repair of normal tissues. The other, it's thought, results in reoxygenation of hypoxic regions in tumors. For example, if you start off with a tumor that has hypoxia and aerobic cells, you radiate, the radiation is going to kill the aerobic cells, it'll leave the hypoxic cells. But over 24 hours, these cells can reoxygenate, and now you treat again, you kill more aerobic than hypoxic, and you keep doing this 35 times, you can get rid of the tumor. So it's thought. This has never been demonstrated clinically. We think this imaging that we have, if we can ever get it into the clinic, will afford us an opportunity to see if this is correct or not. Other means of addressing hypoxia, been about 40 years of development of agents that sensitize hypoxic cells. I showed you an example we did with Dr. Wink, nitric oxide, high LET radiation, hyperbaric oxygen, hypoxic cytotoxins. Hyperbaric oxygen is really a neat one. You put patients 100% oxygen at 3 atmospheres, you force oxygen into the hypoxic regions of the tumor, and then you treat with radiation. In animals, you get a benefit. In people, you do too. But it's so complex to do clinically, and it's also very dangerous to have 3 atmospheres of oxygen around an electrical unit. Okay. So it hasn't been a very popular way of treating cancers with hyperbaric oxygen. What about repopulation? Hopefully, you get it in the esophagus, but not in the tumor. You need repopulation for normal tissues to repair. And this is an example showing that in skin, you can irradiate skin. And at some point, it says, oh my goodness, I've been damaged. I need to repopulate. And so it starts repopulating. And in fact, in order to stop this repopulation, you need to give about an extra 130 rad per day to stop this. So this urge to repopulate is strong. And so I show you an example of repopulation in patients. Patients with head and neck cancer, when you start irradiating the tumor, you're obviously going to irradiate the normal mucosa. And when you do that, after about 2 weeks, the patients get horrendous mucositis. It is really severe. They can hardly swallow. Sometimes they can't eat. And so they beg with the oncologist, please give me a break so this can heal, and then start back again after it's healed. Bad thing. For example, here's patients with head and neck cancer given about the same dose of radiation, 2 gray fractions, but in this case, they split the course. They gave the patient a break of about 10 days. So if you look at the overall time, it's longer, and the 3-year local control is 32%. If you just give it straight through in 5.5 weeks with no break, look at the 3-year local control. It's about double. So you can't break. Even though the normal tissue is proliferating and recovering, so can the tumor. Okay? So it's a delicate balance you play there. Did reassortment occur? And by reassortment, I mean, was there a change in the cell cycle status of proliferating cells in the tumor? The answer is yes, it can. When you irradiate cells that are in mitosis and G2 are most sensitive to radiation, and cells that are in late S and early G1 are most resistant. And in fact, you can get in situations where you give a dose of radiation, you synchronize the cells, and these cells come back around into a radiosensitive phase of the cycle here, and you can sensitize them. It's nearly impossible to do this in the clinic. You can do it in animal tumors. You can irradiate and wait the right amount of time for those cells that were in S phase to come around to mitosis and irradiate. You can get enhancement, but it's too complicated to do. It's easier to use a drug. Okay? One drug that we used in the 1990s prior to clinical trials, a drug called Taxol. Taxol imparts its cytotoxicity a couple of ways, but one thing it does, it blocks cells in G2M, the cell cycle, which is a sensitive phase of the cell cycle. So it's kind of cool. If you look at a survival curve initially, with and without Taxol, there's no difference after, this is a control. If you treat with Taxol for 12 hours, if you look at this BUDR PI flow, you can see the cells in G2M about 50% compared to 12 initially. There's not much enhancement, but if you go 24 hours, you see there are a lot of cells in G2M, 80%, and see you enhance the radiosensitivity with the Taxol. We had cells that are out of proliferation, plateau phase. Log phase you can sensitize, plateaus you can't. So you need cells to be moving if you treat with Taxol so they can block in G2 and be in a sensitive phase for radiation. So we spent a decade on Taxol. We did a lot of cell work in the lab. We did a trial in lung cancer, pancreatic cancer, and head and neck cancer. We got great effects on the tumor in head and neck cancer, but horrendous normal tissue damage, okay, because of proliferating cells in the oral mucosa, okay. Tumor biopsies from 6 patients on prolonged Taxol infusion failed to show an accumulation of G2M. Probably subpopulation was a problem and where you biopsy, but we didn't see any G2M block in the patients. So look at this, great tumor response, the tumor is gone, but look at the normal tissue damage. It's almost like a tornado. This, by the way, is where the beam went through the patient. It's almost like a tornado damage. So great tumor response, but look at normal tissue. The higher the dose, the larger biological effect. Fraction size is very important. If you give high doses per fractions, you will get more normal tissue damage. And that's what this graph depicts. I don't have time to go into it. But it is well known that if you stay with your 2 gray fractions over 5 to 7 weeks, you can avoid a lot of normal tissue damage. If you go to higher fraction sizes, 3, 4, 5 gray per fraction, you're going to buy a lot of late effect normal tissue damage. This is an example of that published several years ago. And I'm showing this because everything I'm talking about, I'm going to reverse about 30 or 40% of it on the next slide, okay? So this is a study where they treated breast cancer patients with cobalt-60. They'd had mastectomy, and they were treating this node region around the arm. And for whatever reason, it's back in the era of cobalt-60, they were using fairly high doses per fraction, all the way up to 5.2 gray. The good news in this clinical trial was that 50% of the patients were alive 30 years post-treatment. Fantastic, of course, if you're in that 50%. But 92% of the long-term survivors had paralysis in their arm, okay? And progression to late effects, some patients, you observed this paralysis in 5 years after treatment, and some went all the way out to 34. So this gift that keeps on giving, fibrosis, it can happen over many years. Of course, it's fortunate if you've got a tumor response and you're alive. I mean, that is one benefit from it, but it's obviously an issue. It led these authors, and a lady I really respect, she passed away a few years ago. Interestingly, she died of breast cancer, Julie Dennekamp. Any tendency toward a return to the use of fractionation schedules with fewer, larger doses for economical reasons in patients with potentially curable disease should be strongly discouraged. Okay? So now what's happening in radiation oncology? We're now giving large doses per fraction, okay? It's been an interesting thing that's happened. The radiation machines have been used for many years, up until the past 5 or 6 years ago, weren't fancy. Okay, they were machines that you could aim the radiation, you could use lead blocks to block normal tissue if you wanted, but mainly AAPA, anterior, posterior, that's it, you're done. Well, physics and companies now have devised machines like this that are really fancy. If you have a 3D image of the tumor, you can have this machine computerized so that it'll go around the patient, and these collimators that are right here will conform to the size, the 3D size of the tumor. So you can deliver a very precise dose to the tumor with minimal dose in normal tissue. And so with that capability, now, if you listen to WTOP coming in the morning, you'll hear Georgetown talking about Gamma Knife or prostate cancer treatment. Oh, it was great. I got my cancer treatment done in 5 days, where it normally would take 5 to 7 weeks. And they pour in, 15, 20, 30 gray per fraction, big doses of radiation, which would give you horrendous fibrosis in normal tissue. But they get away with it because they minimize the normal tissue in the field. You're seeing this now in prostate cancer and in breast cancer and lung cancer, but lung cancer where the tumor is confined to the periphery. They don't want to get near the heart and esophagus and what have you in the center of the body, so they stay with the periphery. They're giving doses out to 15, 20, 30 gray to small volumes in the lung. They're actually using this to ablate tissue. The idea being that in a small volume, you can get away with damaging some of your lung, okay, if that takes care of the tumor. So radiation oncology now is being revolutionized to some extent by these machines. It's really kind of exciting. When I first heard about it 5 or 6 years ago, I was thinking, will be a disaster. This is not a disaster, okay? It's actually working out quite nicely in specific types of tumors. I don't want to take time from Lisa. So I have a whole bunch of stuff I could talk about. I'm not going to do that. We've developed radioprotectors. Maybe I'll take a minute. Chemical radioprotectors, the only FDA-approved agent now that's used in patients is a compound called amifostine. It's an aminothiol. It's administered about 30 minutes before radiation treatment. It's thought that it preferentially protects normal tissue compared to tumor because of compromised uptake in tumor versus normal tissue. It gets into normal tissue faster than in tumor. This is an example where we gave it to mice, C3H mice, 30 minutes before we gave total body radiation at these doses. So animals that get to saline, you see there, this is a survival experiment. We did this very nicely, very humanely. I am vice chair of the NCI ACUC, so we do not do survival experiments anymore. This is, survival to defined euthanasia. That's the correct way of doing it. So you can see animals are very sensitive to the radiation dose, but you give this drug 30 minutes before radiation, and wow, look at that. It is a radioprotector. There have been problems with the drugs in clinical trials. You give it to patients, first thing that happens to them is their blood pressure drops out to wazoo. In fact, the early clinical trials all done in ICU. Couldn't even give radiation, it was so bad. Now there's been some different routes of administration, but there's always an issue and concern, is it protecting the tumor? So it's been used, a lot of clinical trials done, it's not being used very much currently. Now, if you will allow me, I'm going to, so I finish on time, I'm going to go to the last 3 slides. So I'm not going to cover any of this stuff. You have a handout. We developed our own radioprotector in our lab, and one of those slides are about that. In fact, we even found this drug, forget the radiation, it causes you not to get obese, okay? And it's nice for animals. You see a little curve there of some animals on this stuff in the food. And we think we found a mechanism of how it's doing that. It's through the gut microflora, we believe. So it's an interesting story. There's some references there. If you're interested in that kind of stuff, you can look at it. But I want to end up with this last 2 or 3 slides. So what are free radicals? I think you've already had that by other lecturers. When I first came to NCI, this is the paper here I read in 1979 right here by a guy named Britton Chance, first author here. But I think it was the first person to publish that there's leakage of free radicals from the mitochondria. When I read this paper, it's about 30, 40 pages long, I just was blown away because at the time I only thought about free radicals in the context of radiation. I didn't think about them in regard to metabolism. But certainly we know that in the mitochondria, along the electron transport chain, free radicals, superoxide and hydrogen peroxide can be produced. There's always agents to protect it, but it looks like about 2 to 5% of the oxygen consumed results in ROS leakage. That's why people like me get gray-headed prematurely, maybe, in aging. There's a theory along those lines. But at any rate, you're having to deal with oxidative stress daily. And David would also indicate that reactive nitrogen species are also operational here as well. But interesting about nitric oxide, because it's a lot of these things can have 2 faces. They can enhance certain reactions or they can protect against certain reactions. And one of the things that David worked out in our lab is that nitric oxide under certain conditions can actually be an antioxidant and protect against ROS. Kind of interesting. In cancer, we certainly know now there's a lot of oxidative stress going on in cancer cells, which can induce DNA damage and mutation and genomic instability. There's high ROS in cancer cells, which can accelerate pro-survival pathways. That's pretty well documented now. And certainly in cancer cells, there may be altered levels of antioxidants. In fact, Lisa trained under a fellow who started this ball rolling in 1979 in cancer research, Larry Oberle, in regard to superoxide dismutase in cancer, is a classic paper. Various chemotherapy drugs that we use, and I say we use because we still use them. I can tell you, I had a son with cancer a few years ago, and he got this drug, and this drug, and this drug. And you know what? It worked. He's 5 years out right now. So I don't ever underplay cytotoxic chemotherapy drugs. They're awful, okay? They have terrible side effects, but under certain circumstances, they can work. And these in particular work by free radical mechanisms. We show that if you take bleomycin, for example, and you take glutathione out, you sensitize to bleo. You increase glutathione with oxythiazolidine, you can protect. Same with melphalan. Neocarcistatin is a weird drug. It requires thiols to be activated to a cytotoxic agent. So if you take glutathione out, you don't get any killing at all. Kind of a cool drug. There's solid information now that these, what we always call alkylating agents, causing DNA damage, can also induce ROS. So free reactive oxygen species and free radicals are here to stay. And they're certainly present in cancer cells. So this patient that I started with, I want to end this story. I got these CT scans from a friend of mine at the University of Pennsylvania. This patient died 1 year after radiation treatment. She died of metastatic disease, probably because the drug penetration of the drug was not good in the Mets. If you want to read about the problems of trying to get a drug to tumor, read this paper here by Ian Tannock, Drug Penetration in Solid Tumors. I think you'll be surprised if you haven't read this. Interestingly, though, other patients can present with non-small cell lung cancer. That's it right here. Looks pretty bad. They get treated with same fractionation the lady before was treated, same total dose, and they're disease-free at 5 years. That's some normal tissue damage. The tumor's gone. Why is that? This patient didn't have metastasis, okay? So local control radiation works pretty well. Metastasis is a challenge. And the hope is that some of you young folks, as you continue your career, will figure out how to make the previous patient survive. So I'm going to stop. I'm pretty much on time, and sorry I didn't get over the whole thing. It's because I was sitting, that's why. Yes. The answer is yes. The repair is completed, as I indicated, in about 5 to 6 hours. You irradiate again, will undergo the same process. They can undergo, tumor cells and normal tissue can undergo repeated cycles of repair. I actually, as a graduate student, proved that to myself many years ago. No, the rates are the same.
So our next speaker is Lisa Ridner, and she works with David Wink, so she gets to see him all the time and hear about nitric oxide. And she's a member of our redox biology faculty, and she's with the radiation biology branch. She's going to talk about mechanisms of NOS2-associated breast cancer progression.
Okay. All right, so Dave and I have been studying roles for NOS-related mechanisms in cancer progression for a few years now. We've been looking at breast cancer because we've been involved with Stefan Ams now for several years in an ongoing collaboration. And Stefan has a breast tumor cohort from about 250 breast cancer patients where we have examined various signaling pathways through immunohistochemistry staining for these proteins. And we have correlated the effects of the expressions of these proteins on patient survival. And so that is going to be a large part of what I talk today about, data that we have accumulated using this patient cohort. So to date, let's see. Okay. Breast cancer remains the second leading cause of death among women with malignancies. Currently, classical prognostic indicators to stratify and predict therapeutic response and survival of patients include clinical biomarkers, including lymph node metastases, tumor size, tumor grade, age, peritumoral vascularization. Also, biological markers including estrogen receptor, progesterone receptor, HER2 new expression, and P53 are also parameters that are employed to predict survival. Okay, but for improvement of breast cancer management and to avoid both under or over treatment and improve quality of life and reduce the cost of the treatment, this process, warrants the identification of new biomarkers that will aid in predicting patient therapeutic response and survival. So inflammation has been long known to be associated not only with the development of cancer, but also the progression of cancer. And so post-tumor interactions predict cancer progression and patient survival in this case. So, and why is that? Under normal conditions, when a tissue is injured or exposed to a pathogen, it will elicit an immune response, and a number of immune cell mediators will infiltrate the area, and it will kill the pathogen, for example, and then it will begin to heal. Immunosuppression will begin, and you'll get the infiltration or secretion of cytokines that promote wound healing and neovascularization in the wounded tissue, and then you get tissue restoration and healing, and the inflammatory process goes away. But during carcinogenesis, you have chronic inflammation, and you get a lot of epithelial cell mutations and hyperproliferation and ongoing inflammation. And this results in the development of loops that promote immunosuppression and the influx of immune cell mediators that are primed to secrete cytokines and growth factors and different things that promote chronic inflammation and sort of like a wound healing response that never goes away. And then you get tumor cell survival and metastasis associated with cancer progression. So we're interested in looking at markers of inflammation during cancer progression. And one of those markers is the inducible form of nitric oxide synthase. We're also interested in looking at toll-like receptors because toll receptor signaling can also promote tumor cell survival and metastases, and is also resistance to drug therapies. And we're also interested in looking at components of the extracellular matrix that can be involved in these processes, including MMPs and TIMPs. So nitric oxide effects are concentration dependent. They're also temporally dependent and spatially dependent. But we know from probably at least 10 years of studies that low levels of nitric oxide are involved in normal cell processes, including angiogenesis. They're cyclic GMP dependent, and they can occur at levels of NO as low as or less than 2 nanomolar in NO flux. And then as the concentrations of NO increase, you can get pro-survival signaling around 300 nanomolar of steady state NO. You can get the upregulation of molecules that predict poor cancer survival, including CD44, p-cadherin, and COX-2. And at higher levels, you can get p53 phosphorylation. You can also get the induction of p53 mutations, which we're looking at now, and other mutations. And that occurs at higher levels of nitric oxide. Okay, so NOS2 is involved in regulation of many processes, including inflammation, wound healing, angiogenesis, proliferation, and survival, and cancer survival. And interestingly, MMPs regulate many of the same processes, They're involved in the development and differentiation pathways, wound healing, inflammation, angiogenesis, and cancer. Under normal conditions, they're very tightly regulated, but when that regulation is lost, they can go on and they can continue to chew up matrix, extracellular matrix, in a manner that is not good, and it can, for example, promote cancer cell metastases and migration, and so as well as neovascularization and things that you don't really want going on in a tumor, because that will promote its survival and progression. TIMPs are endogenous inhibitors of MMP that can also modulate cell growth and survival. TIMPs will bind directly to an active MMP and basically block its ability to bind to a substrate and cut it, okay? But they also have MMP-independent signaling capabilities, and they can, for example, upregulate pro-survival signaling through PI3 kinase and AKT and downstream phosphorylation of fat to inhibit the bad and inhibit its pro-apoptotic effects. So I'm going to go on now and talk about how NOS2 promotes breast cancer progression. This is a study that Sharon Glenn published in the Journal of in JCI in 2010. And in this study, she showed that the breast cancer patients that had high NOS2 demonstrated very low levels of survival, but only in ER-negative breast cancer. This did not happen. This was not observed in ER-positive breast cancer. And of course, ER-negative is a more aggressive form. So this is very important data showing that NOS2 is associated with the development of more aggressive cancer phenotypes. So also there's prognostic significance of MMP9 and TIMP1 as being predictors of poor survival in breast and other types of cancers. And this slide shows that patients that have high MMP9 or high TIMP1 demonstrate both reduced levels of overall survival and relapse-free survival. And that's kind of interesting when you think that TIMP1 is an inhibitor of MMP9. Why would patients that have high TIMP1 have worse survival? So we asked the question whether or not nitric oxide could shed some light on the pro-survival effects of MMP9 and TIMP1. So several years back, I did a series of experiments where I looked at regulation of MMP9 in murine macrophages. And what we found was that NO could regulate MMP activity, MMP9 activity, in two different mechanisms. Under low flux NO, you got increased MMP9 activity, which was cyclic GMP dependent, and it functioned by reducing TIMP1 protein secretion. But at higher levels of NO, you got a direct activation of the MMP9 protein that occurred around about 5 to 10 times, at a 5 to 10-fold higher flux than this cyclic GMP-dependent process. And as the NO levels continued to rise, it would also inhibit the activity of the enzyme. So it was kind of a biphasic response. Okay, so we wanted to go on and examine this relationship in a physiologically relevant model. So we looked at it in human breast tumors. And what we did was we stained the breast tumors for TIMP1. And you see this panel reflects high TIMP1 staining, moderate TIMP1 staining, and no staining in breast tumors, in breast tumor tissue. And CD63 is a cell surface protein that TIMP1 can bind to initiate pro-survival signaling. So we also looked at CD63 in these tumors, and again, we show various levels of staining in the tumors, from high staining to moderate staining and, little to no staining. So we wanted to correlate survival effects. And we found that TIMP1 in our breast tumor cohort did predict poor survival if it was expressed at high levels. And so then we went on and stratified for the effects of NOS2. And what we found was that this predictive effect of TIMP1 seemed to be restricted only to tumors that expressed high NOS2. And as tumors that expressed low NOS2 had no predictive ability on survival. So these results suggested that the survival effects of high NOS2 and TIMP1 may be mechanistically interrelated. So then again, as I said earlier, TIMP1 has MMP-independent functions. It can bind to CD63 to initiate FAK PI3 AKT signaling that culminates in phosphorylation and inhibition of BAD, okay? And so we looked at this relationship in our breast tumor cohort. And earlier, Robin Pruitt had demonstrated that there was an association between NOS2 expression and AKT phosphorylation in these patients, as evidenced by an odds ratio of about 4.5. When we stratified for high versus low TIMP1 expressing tumors, we found that this odds ratio went from 4.5 in all patients up to about 13 in patients whose tumors also expressed high TIMP1. Interestingly, patients whose tumors expressed low TIMP1 had a reduced odds ratio. So this suggested that TIMP1 could indeed affect the relationship between NOS2 and AKT phosphorylation in these breast tumors. So we then went on to look at, correlations between NOS2 and TIMP1. And what we found was that we only found a positive correlation between NOS2 and TIMP1 in ER-negative patients. So this suggested a potential role in patients who had more aggressive ER-negative breast tumors. So we went on and explored this possibility in ER-negative breast cancer cells. And to do that, we used INO-treated cells that had been pretreated with a morpholino, an antisense morpholino, which blocked the translation of TIMP1 protein. And it did so pretty effectively out through 72 hours, as you can see in the top panel. So in these cells that were pretreated with TIMP1, we then exposed them to increasing concentrations of the INO donor DTANO, and we looked at AKT phosphorylation and BAD phosphorylation. And what you can see is the cells that had suppressed TIMP1 levels had reduced INO-induced PAKT and reduced INO-induced phosphorylation of BAD. And that is plotted in the lower panels of this slide. So you can see a pretty dramatic effect on TIMP1 suppression, abating these phosphorylation signaling cascades. So mechanistically, we wanted to know if INO enhanced TIMP1. And we looked at real-time expression and really didn't see any effect on TIMP1 message. And we saw a significant increase in TIMP1 protein secreted into the media, but this occurred at 100 micromolar of DTANO, whereas the pro-survival signaling effects that I just showed you occurred at a higher concentration, 500 and 1,000 micromolar of DTANO. So we did not know what the mechanism was at this point. An interesting paper came out showing that TIMP1 contributes to ovarian anomalies, and what they looked at was mutant TIMP1, which lacks MMP inhibitory function, and they demonstrated in this paper that this mutant form of TIMP protein actually bound to the CD63 protein to a higher degree when compared to the wild type protein. So this suggests that a TIMP1 protein that's lost its ability to inhibit active MMPs is getting turned over, or at least it's binding to the CD63 protein at a higher degree. Okay, and also they demonstrated the inhibition of apoptosis in this model in association with the mutant TIMP protein. So we asked the question whether or not NO could modulate TIMP1 CD63 colocalization in the breast cancer cells and in the breast tumor. So to do this, we did a confocal study, and we exposed the cells to Detaino, and then we we looked at immunofluorescing TIMP, which fluoresced in green, as well as CD63, which fluoresced in red. And what you can see is at the high concentrations of the NO donor Detaino, you see a lot more green. But you also see more co-localization of the two proteins, as evidenced by these yellow-orange areas on the insets here. So these results suggested that NO could modulate the co-localization of TIMP1 to the CD63 cell surface protein. So how might that occur? NO can induce nitration events. So we looked at whether or not NO could nitrate TIMP1. And to do this, we used recombinant TIMP1 protein exposed under the same conditions to the same NO concentrations. And what we found was a nice 45 Dalton mass shift, which is indicative of a nitration event in the NO-treated protein. And the nitration events were identified at two specific tyrosine residues by mass spec at residue 95 and 143 in the TIMP1 protein. So we then went on and looked at immunoprecipitated TIMP1 in our cells that were exposed to NO, and we found that when we IP'd out TIMP1 and blotted for 3 and T, we see higher levels of nitration in the NO-treated cells, and we see also co-localization of CD63 on the immunoblots. So these results suggest that NO can modulate CD63-TIMP1 interaction. And because we previously looked at AKT phosphorylation and downstream phosphorylation of BAD, we then wanted to go back and look at upstream kinases, or at a kinase that's upstream of AKT. So we looked at PI3 kinase in the TIMP1 knockdown study, and we found that at 500 micromolar Detaino, TIMP1 suppression dramatically reduced the phosphorylation of PI3 kinase at this dose of NO. So in conclusion, the role of TIMP1 in breast tumor biology, TIMP1 predicts poor breast cancer-specific survival in a patient cohort. It is capable of augmenting NOS2 AKT association in these breast tumors. And in breast cancer cells, if you silence TIMP1, you can also induce or reduce NO-induced PI3, AKT, and BAD survival signal. So what about the effects of NO on MMP9 and MMP9-TIMP1 ratio in breast cancer? So the nitration story is still pretty interesting because about the same time that we published that paper, another group came out and showed that when they induced nitration events in their macrophage model, they were able to show that TIMP1 that was nitrated could not bind and inactivate MMP9. And they did this in LPS-stimulated macrophages, which induces a lot of NO, and they used a reverse zymography. So what this shows is that in this, the cells that got LPS, you don't see TIMP1 binding to MMP9, and you see higher MMP9 activity. So they actually used a math model to predict which tyrosine residues were being nitrated. And interestingly, the tyrosine residues that this math model predicted were the same residues that we showed by mass spectrometry on the previous slide. So I think that this is pretty convincing data showing that NO can modulate these processes through nitration events of TIMP1 protein. It can inhibit its ability to block MMPs. So in a cancer microenvironment, you're going to have more MMPs that can facilitate cell migration and metastasis. On the flip side of the coin, you can have a nitrated TIMP molecule that can induce pro-survival signaling of that cancer cell. So we also went back to our cohort and we looked at MMP9 predicting survival in these patients, and we found that high levels of MMP9 do predict poor survival in these patients. And under low TIMP conditions, the hazard ratio is increased, okay? But under high conditions of TIMP1, MMP9 did not predict poor survival in this patient cohort. So again, I think that's pretty interesting, implicating a role for NO in the development of more aggressive phenotypes and cancer progression in this cohort. So what's the effect of MMP9 in the tumor microenvironment? It can promote angiogenesis. It can be involved in cytokine processing, making them more bioavailable or potentiating their biological activity just by cleavage events. It can promote cell migration and invasion. And going back to this paper that we published in PNAS in 2007, this shows a role for NO-induced MMP9 in pro-angiogenic response. And this is an implant model where we used a plug of tissue implanted in an agar matrix, and then you could treat it with various angiogenesis-inducing reagents, and then you would look at the number and distance of endothelial cells that would migrate away from the edge of tissue. And so what this panel shows is that when we took the conditioned media from NO-treated macrophages and we put it on this plug of tissue, we saw enhanced levels of endothelial cell migration away from the edge of the plug of tissue when compared to the control that just got media alone, okay? And this is quantified in panel B on the slide. And interestingly, when we went back and silenced MMP9 in this experiment, we were able to abate that proangiogenic process induced by NO. So we wanted to know if NO had a role in increasing MMP9 secretion from breast cancer cells. So we looked at NO-treated MCF7 cells, and we saw increased MMP9 at the higher doses of the exogenous donor Detaino. We also showed it in MB468 cells and in MB231 cells, which are ER-negative breast cancer cells. So the next experiment that we did was we did an in vitro angiogenesis assay where we plated endothelial cells on a matrix, and then you can put different drugs or growth factors, whatever, in the media, and you look at these branching structures here, as well as the outgrowth of capillary structures of the endothelial cells. And so when we took conditioned media from MB231 cells that were not treated, we saw a slight increase in, the formation of these branching structures, okay? But when we treated the MB231 cells with NO and took that conditioned media and put it on the in vitro angiogenesis, we see a dramatic increase in the enclosed structures as well as the branching of the endothelial cells. So this is telling us that NO is inducing a proangiogenic response of the tumor cells themselves. So the next thing that we have to do is repeat this experiment under conditions of MMP9 silencing to see if we can tease out a specific role for MMP9 in this process. So as I stated earlier, MMP9 can process cytokines, making them more biologically active. Actually, it does that with VEGF. It can cleave it from the being bound to the matrix. It can cleave it and release it to make it more bioavailable. With IL-8, it can truncate the protein and make it and potentiate its bioactivity. This is some work that Sharon published in her JCI paper, and this simply shows that NO can increase the secretion of IL-8 from MB231 cells and MB468 cells. These are the aggressive ER-negative cells. She didn't see this effect in ER-positive cells, which are less aggressive. Okay. As I said earlier, MMP9 can truncate IL-8, which translates to a potentiated ability of it to bind its cell surface receptors. So this is the control protein, this is the truncated protein, and you can see that the curve shifts, pretty significantly to the left. IL-8 also is involved in migration. It can induce angiogenesis and cell migration as well. So Sharon looked at both migration and invasion of MB231 cells and MB468 cells exposed to NO, and you see an increasing dose-dependent response of migration and invasion of these cells when they see NO. So some of the proteins that are upregulated during this process are CD44, as well as PCAD-herin and S100A8. But regarding CD44, it can serve as a docking port for MMP9. And at the leading edge of a migrating cell, CD44 will bind MMP9, and then MMP9 can chew through matrix, allowing the cell to move through the matrix. So that's another potential mechanism where NO could modulate, a more aggressive response of a tumor. So this is just a summary slide showing different mechanisms involving these matrix proteins where NO can activate MMP9 and affect IL-8 and VEGF signaling, as well as motility and metastasis through docking with CD44. It can also nitrate TIM1, which inhibits its ability to inactivate an MMP, but potentiates its ability to bind to CD63 and promote pro-survival signaling. So I'm just going to finish up with some more recent findings that we have been able to identify regarding mechanisms of inflammation in cancer. So as I said earlier, when the inflammation process kind of runs amok, it actually will promote cancer survival and progression rather than get rid of the tumor. And it can do that by increasing the levels of immune cell mediators, including undifferentiated myeloid-derived suppressor cells, an influx of tumor-associated macrophages, as well as T regulatory cells and immunosuppressive T cells. And those immune cell mediators can actually promote the progression of more aggressive cancer phenotypes by, secreting cytokines and growth factors and things like that the tumor will actually use to survive and metastasize. So again, this is our slide showing that high levels of NO predict poor survival in ER-negative cancer patients. We used laser capture microdissection to identify a gene signature of high versus low NOS2 expressing proteins, and this is a host of different genes that are differentially regulated depending upon high versus low NOS2 expression. But the one that we're interested in talking about right now is S100A8, which is five-fold enhanced in these tumors. These are the breast tumors that we examined. And S100A8 is an agonist for toll-like receptor 4, okay? It activates toll-like receptor 4, and when expressed on tumors, this can be associated, activation of TLR4 can be associated with poor survival response and poor drug response of the tumor. So with all of these things in mind, and as I've told you, NOS2 is associated or a predictor of poor survival in breast cancer and many other types of cancers, the question still remained how NOS2 is regulated in a tumor microenvironment. So that we know that a fast-growing tumor is going to use up nutrients and oxygen, and it becomes hypoxic and nutrient deprived. And these kinds of conditions will induce an inflammatory response that, through selection, will cause the development of more aggressive tumor phenotypes. So in the next study that I'm going to show you, we sort of mimicked these components of a tumor microenvironment to study NOS2 expression in breast cancer cells. So the very first piece of data that I'll show you are nude mice that we implanted MB231 ER-negative breast cancer cells that were tagged with fluorescing GFP protein. Okay, so we monitored the growth of these tumors in animals that got regular drinking water, but also got the NOS2 inhibitor aminoquinidine in their drinking water. And what you can see is nearly a 60% reduction in tumor size in the animals that were NOS inhibited. That's quantified here on the left, but it's also qualified by imaging of the fluorescing tumors in the mammary tissue of these animals. So, metastasis is what kills you, right? So we wanted to look at brain mets and whether or not NOS2 inhibition would affect brain mets. And this panel shows a dramatically reduced level of fluorescing protein in the NOS-inhibited brain when compared to control. And when we quantified this by real-time analysis of the GFP tag, we see about a 90-fold reduction in mets in the brain in animals that were NOS inhibited. So we went back and we looked at a number of markers of tumor progression in these NOS-inhibited tumors, and we saw that they were all inhibited, or they were all reduced in the NOS-inhibited tumor. Similarly, we looked at migration of NOS-inhibited MB231 cells, and you can see when compared to the black line, the NOS-inhibited cells didn't really move at all. And this was reversed when we titrated exogenous NO donor back into the cells. Okay. Also, we looked at the effect of NOS2 expression on Taxol toxicity of these breast cancer cells. And to do this, we used serum withdrawal coupled with interferon gamma stimulation, which we show enhances NOS2 expression in these tumors. And what we see is that the serum withdrawal increased the survival of cells that got Taxol when compared to the untreated control. And even though in this experiment, aminoguanidine did not blunt the effect, but when we added 100 micromolar of the exogenous NO donor to these cells, we actually potentiated it, okay? So this is an experiment where we looked at NOS2 expression under conditions of serum withdrawal. And you see a lot of NOS2 expression in these cells. When we treated them with aminoguanidine, the expression levels went up a lot, okay? When she hit them with when she hit the gamma-treated cells with aminoguanidine, she didn't really see much of a difference in the fluorescence levels, but when she treated just the serum withdrawal treated cells with aminoguanidine, it dropped the basal levels of NO expression. So in this panel, again, she looked at serum withdrawal, and this is a quantified slide. So basically, we show that serum withdrawal induces NOS2 expression when compared to the control. It can be evaded by aminoguanidine, but reaccumulate by exogenous NO donor. So these experiments suggest that basal NOS2 derived NO can mediate feed-forward NOS2 protein regulation in breast tumors and breast tumor cells. So then she looked at S100A8 expression under the same conditions, and she sees dramatic induction by gamma interferon in serum withdrawal, which is abated by aminoguanidine, and she can reverse this process by titrating back exogenous NO. The TLR4 expression is not so much affected by gamma, but if she adds aminoguanidine to the cells, she can reduce the TLR4 expression in these cells. So IL-6 is a downstream target of S100A8 activation of TLR4 signaling, and she shows that gamma induces IL-6, which is abated by NOS2 inhibition. And similarly, TIMP1 is also upregulated by gamma stimulation, but it's abated by aminoguanidine. So the next thing that we wanted to do was look at NF-kappa B regulation under these conditions. NF-kappa B, it has been found in the promoter regions of many of the genes that are upregulated by NOS2 in this breast tumor cohort. And NF-kappa B is also a key mediator of toll receptor signaling. Okay, so Chris Switzer did this work, and the same mouse tumors that I showed you in the previous slides, he looked at phosphorylation of the P65 subunit of NF-kappa B in control and aminoguanidine tumors, and he sees that the NOS-inhibited tumors demonstrate a significant reduction in P65 phosphorylation, and also they demonstrate a significant reduction in the IL-6 protein levels. So he went back and he did similar experiments using NOS2 transvected MB468 cells and MB231 cells, and he shows that when he adds aminoguanidine into the mix, he can reduce NF-kappa B activation in these cells. And similarly, he can induce NF-kappa B activation just by titrating in the NO donor. So again, IL-6 levels are induced by NO in these cells, but they're abated by both a PI3 kinase inhibitor and an NF-kappa B inhibitor. And this figure shows that the NF-kappa B activity is also dependent on S100A8 and TLR4, because when he uses silencing technology to knock them down, he cannot induce NF-kappa B activation by NO. Same thing is shown here again with IL-6. The upregulation of IL-6 is also S100A8 dependent. So he went back to our tumor cell cohort and he looked for a correlation between IL-6 and S100A8 in these patients, and he found that it was restricted to high NOS2 expressing tumors, as you can see by our p-value of 0.02. And also it was restricted to ER-negative tumors, suggesting that it occurred only in the more aggressive tumor phenotypes. So this summary is just kind of a schematic to show that NOS2 expression in the tumor microenvironment can promote a feed-forward chronic inflammation that continues to perpetuate the NO levels that will mediate pro-survival signaling, as well as chemoresistance and cell migration, tumor growth, and metastases. So these are the people that were involved in all of this work. Sharon Glenn and Stefan Ams were involved with the analysis of the breast tumors. Yong Lee and Harris Wyfantis were the pathologists that scored immunohistochemistry staining of the tumors. Mike Lizardo did confocal. Ernie Brueggemann did our mass spectrometry. And these are the students in the Wink lab that did the rest of the work. So thanks. Well, NOS2 definitely is an inflammatory mediator, and NOS2 can be associated with tumor cell killing. But again, it goes back to the concentrations of NO that are generated, and NOS2 can produce a range of nitric oxide flux depending upon the stimulant. Also, NO can react with superoxide. So the presence of superoxide can affect the bioavailability of NO that is present to conduct various, signaling pathways that we've talked about. It's also a gas, so it can diffuse, okay? It can diffuse away from the cell. So all of these factors are associated with the response of NO under these conditions. But we do know that if you really hit with a high level of NO, you can kill the tumor, because we have other data where we have used NO donors that are conjugated to aspirin. And we used that same mammary tumor model with the GFP fluorescing MB231 cells, and we gave them Dino aspirin, and Dino will release the NO very, very quickly, like within 2 minutes, whereas Detano releases its NO over a longer period of time, 20 hours, at a slower rate, okay? So when you put the Dino aspirin in these animals and it releases a ton of NO, it did kill the tumor. We used another donor called Ipano, which is a very interesting molecule. It can release nitric oxide, but it can also release HNO, which is a reduced form of NO. And that was very, very effective at killing the tumors. So, different things happen depending upon what species you're talking about, but also the concentration of the donor will affect different processes itself. As far as the toll receptor signaling, yes, toll receptor signaling, it can also kill tumors as well. In fact, 100, 200 years ago, a physician named William Coley would inject bacterial proteins into tumors that would initiate a profound immune response and he would kill 30% of the tumors. So yes, it can do both, but depending upon the specific conditions, and again, the immune cell mediators, if you have a whole bunch of tumor-associated macrophages floating around, that's not good. If you have a bunch of Tregs, in the tumor microenvironment, that's going to be immunosuppressive. So in terms of therapeutic response, you want to abate immunosuppression. That's what, like the Coley's toxin would do. Nowadays, they use a molecule called BCG, I think. Is that right? Basically, it's just a peptide, like a bacterial peptide, and they give it with the various chemotherapeutic drugs, and I think they've also given it with radiation. And they have improved the therapeutic response of these different therapies. And one thing that it does is it blunts immunosuppression. And one mediator of immunosuppression that is affected is IL-10. If you knock down IL-10, which is an immunosuppressor, you can improve your immune response and the therapeutic response as well. So. Okay.